Welcome to our video on lymphatic system. What is a lymphatic system? What its use in our body? The lymphatic system, or lymphoid system, is an organ system in vertebrates that is part of the immune system and complementary to the circulatory system. It consists of a large network of lymphatic vessels, lymph nodes, lymphoid organs, lymphoid tissues and lymph. Unlike the circulatory system that is a closed system, the lymphatic system is open. The human circulatory system processes an average of 20 liters of blood per day through capillary filtration, which removes plasma from the blood. Roughly 17 liters of the filtered blood is reabsorbed directly into the blood vessels, while the remaining 3 liters are left in the interstitial fluid. One of the main functions of the lymphatic system is to provide an accessory return route to the blood for the surplus 3 liters. Lymph is a clear fluid carried by the lymphatic vessels back to the heart for recirculation. The Latin word for lymph, lympha, refers to the deity of fresh water. Lymph, from Latin, lympha, meaning water, is the fluid that flows through the lymphatic system, a system composed of lymph vessels, channels, and intervening lymph nodes whose function, like the venous system, is to return fluid from the tissues to be recirculated. At the origin of the fluid return process, interstitial fluid, the fluid between the cells in all body tissues, enters the lymph capillaries. Structure of lymph has a composition similar but not identical to that of blood plasma. Lymph that leaves a lymph node is richer in lymphocytes than blood plasma is. The lymph formed in the human digestive system called chyle is rich in triglycerides, fat, and looks milky white because of its lipid content. The structure of lymphatic system consists of a conducting network of lymphatic vessels, lymphoid organs, lymphoid tissues, and the circulating lymph. The primary, or central, lymphoid organs generate lymphocytes from immature progenitor cells. The thymus and the bone marrow constitute the primary lymphoid organs involved in the production and early clonal selection of lymphocyte tissues. The thymus is a specialized primary lymphoid organ of the immune system. The thymus is located in the upper front part of the chest, in the anterior superior mediastinum, behind the sternum, and in front of the heart. It is made up of 2 lobes, each consisting of a central medulla and an outer cortex, surrounded by a capsule. In children, the thymus is pinkish-gray, soft, and lobulated on its surfaces. At birth it is about 4 to 6 centimeters long, 2.5 to 5 centimeters wide, and about 1 centimeter thick. It increases in size until puberty, where it may have a size of about 40 to 50 grams, following which it decreases in size in a process known as involution. The arteries supplying the thymus are branches of the internal thoracic and inferior thyroid arteries, with branches from the superior thyroid artery sometimes seen. The branches reach the thymus and travel with the septa of the capsule into the area between the cortex and medulla, where they enter the thymus itself, or alternatively directly enter the capsule. The veins of the thymus, the thymic veins, end in the left brachiocephalic vein, internal thoracic vein, and in the inferior thyroid veins. Sometimes the veins end directly in the superior vena cava. Lymphatic vessels travel only away from the thymus, accompanying the arteries and veins. These drain into the brachiocephalic, tracheobronchial and parasternal lymph nodes. The nerves supplying the thymus arise from the vagus nerve and the cervical sympathetic chain. Branches from the phrenic nerves reach the capsule of the thymus, but do not enter into the thymus itself. The thymocytes and the epithelium of the thymus have different developmental origins. The epithelium of the thymus develops first, appearing as 2 outgrowths, one on either side, of the 3rd pharyngeal pouch. It sometimes also involves the 4th pharyngeal pouch. These extend outward and backward into the surrounding mesoderm and neural crest-derived mesenchyme in front of the ventral aorta. Here the thymocytes and epithelium meet and join with connective tissue. The pharyngeal opening of each diverticulum is soon obliterated, but the neck of the flask persists for some time as a cellular cord. By further proliferation of the cells lining the flask, buds of cells are formed, which become surrounded and isolated by the invading mesoderm. The epithelium forms fine lobules, and develops into a sponge-like structure. During this stage, hematopoietic bone marrow precursors migrate into the thymus. Normal development is dependent on the interaction between the epithelium and the hematopoietic thymocytes. Iodine is also necessary for thymus development and activity. Functions of thymus gland: T-cell maturation, positive selection of T-cell distinct T-cell receptors. Negative selection and TCR gene rearrangements. T cells that attack the body's own proteins are eliminated in the thymus, called negative selection. Immunodeficiency due to failure of thymus development relating congenital problems. Autoimmune disease due results from a genetic defect of the thymus tissues and also in autoimmune regulator, AIRE, gene and cancers. Bone marrow is a semi-solid tissue found within the spongy, also known as cancellous, portions of bones. In birds and mammals, bone marrow is the primary site of new blood cell production, or hematopoiesis. It is composed of hematopoietic cells, marrow adipose tissue, and supportive stromal cells. In adult humans, bone marrow is primarily located in the ribs, vertebrae, sternum, and bones of the pelvis. Bone marrow comprises approximately 5% of total body mass in healthy adult humans, such that a man weighing 73 kilograms, 161 pounds, will have around 3.7 kilograms, 8 pounds, of bone marrow. The composition of marrow is dynamic, as the mixture of cellular and non-cellular components, connective tissue, shifts with age and in response to systemic factors. In humans, marrow is colloquially characterized as red or yellow marrow depending on the prevalence of hematopoietic cells versus fat cells. For example, a newborn baby's bones exclusively contain hematopoietically active, red, marrow, and there is a progressive conversion towards yellow marrow with age. In adults, red marrow is found mainly in the central skeleton, such as the pelvis, sternum, cranium, ribs, vertebrae and scapulae, and variably found in the proximal epiphyseal ends of long bones such as the femur and humerus. In circumstances of chronic hypoxia, the body can convert yellow marrow back to red marrow to increase blood cell production. Human marrow produces approximately 500 billion blood cells per day. Marrow contains hematopoietic stem cells which give rise to the three classes of blood cells that are found in circulation: white blood cells, leukocytes, red blood cells, erythrocytes, and platelets, thrombocytes. Functions of bone marrow. In a bone marrow transplant, hematopoietic stem cells are removed from a person and infused into another person, allogenic, or into the same person at a later time, autologous. If the donor and recipient are compatible, these infused cells will then travel to the bone marrow and initiate blood cell production. Transplantation from one person to another is conducted for the treatment of severe bone marrow diseases, such as congenital defects, autoimmune diseases or malignancies. Before radiation therapy or chemotherapy in cases of cancer, new stem cells are introduced. The earliest fossilized evidence of bone marrow was discovered in 2014 in Eusthenopteron, a lobe-finned fish which lived during the Devonian period approximately 370 million years ago. Study the fossilized interior of the skeleton's humerus, finding organized tubular structures akin to modern vertebrate bone marrow. Eusthenopteron is closely related to the early tetrapods, which ultimately evolved into the land-dwelling mammals and lizards of the present day by scientists from Uppsala University and the European Synchrotron Radiation Facility. The secondary, or peripheral, lymphoid organs, which include lymph nodes and the spleen, maintain mature naive lymphocytes and initiate an adaptive immune response. The secondary lymphoid organs are the sites of lymphocyte activation by antigens. Activation leads to clonal expansion and affinity maturation. Mature lymphocytes recirculate between the blood and the secondary lymphoid organs until they encounter their specific antigen. The spleen is an organ found in almost all vertebrates. Similar in structure to a large lymph node, it acts primarily as a blood filter. The spleen is underneath the left part of the diaphragm and has a smooth, convex surface that faces the diaphragm. It is underneath the 9th, 10th, and 11th ribs. The other side of the spleen is divided by a ridge into two regions, an anterior gastric portion and a posterior renal portion. The gastric surface is directed forward, upward, and toward the middle, is broad and concave, and is in contact with the posterior wall of the stomach. Below this it is in contact with the tail of the pancreas. There are 4 ligaments attached to the spleen: gastrosplenic ligament, splenorenal ligament, colicosplenic ligament, and phrenolochic ligament. Near the middle of the spleen is a long fissure, the hilum, which is the point of attachment for the gastrosplenic ligament and the point of insertion for the splenic artery and splenic vein. There are other openings present for lymphatic vessels and nerves. Like the thymus, the spleen possesses only efferent lymphatic vessels. The spleen is part of the lymphatic system. Both the short gastric arteries and the splenic artery supply it with blood. The germinal centers are supplied by arterioles called penicillary radicals. The spleen is innervated by the splenic plexus, which connects a branch of the celiac ganglia to the vagus nerve. The underlying central nervous processes coordinating the spleen's function seem to be embedded into the hypothalamic pituitary adrenal axis and the brainstem, especially the subfornical organ. The spleen is unique in respect to its development within the gut. While most of the gut organs are endodermally derived, the spleen is derived from mesenchymal tissue. Specifically, the spleen forms within and from the dorsal mesentery. However, it still shares the same blood supply, the celiac trunk, as the foregut organs. The red pulp of the spleen is composed of connective tissue known also as the cords of Billroth and many splenic sinusoids that are engorged with blood, giving it a red color. Its primary function is to filter the blood of antigens, microorganisms, and defective or worn-out red blood cells. 76 to 79% of a normal spleen is red pulp. Unlike white pulp, which mainly contains lymphocytes such as T cells, red pulp is made up of several different types of blood cells, including platelets, granulocytes, red blood cells, and plasma. The red pulp also acts as a large reservoir for monocytes. These monocytes are found in clusters in the Billroth's cords, red pulp cords. The population of monocytes in this reservoir is greater than the total number of monocytes present in circulation. They can be rapidly mobilized to leave the spleen and assist in tackling ongoing infections. The splenic sinusoids are wide vessels that drain into pulp veins which themselves drain into trabecular veins. Gaps in the endothelium lining the sinusoids mechanically filter blood cells as they enter the spleen. Worn-out or abnormal red cells attempting to squeeze through the narrow intercellular spaces become badly damaged and are subsequently devoured by macrophages in the red pulp. In addition to clearing aged red blood cells, the sinusoids also filter out cellular debris, particles that could clutter up the bloodstream. Blood in the arteries end in the Billroth's cords, red pulp cords. These cords are made up of fibroblasts and reticular fibers which form an open blood system without an endothelial lining, and it is within these cords that F480 plus macrophages are found, which are associated with the reticular cells of these areas and are collectively known as red pulp macrophages. From the Billroth's cords, the blood passes into the venous sinuses of the red pulp, which are lined with discontinuous endothelium as well as stress fibers extending under the basal plasma membrane, parallel to the cellular axis. This arrangement of the stress fibers combined with the parallel arrangement of the sinus endothelial cells forces the blood in the red pulp through slits that are formed by the stress fibers. However, this passage can become difficult for aging red blood cells due to their less flexible membranes and therefore they get stuck in the cords and they will be subsequently phagocytosed by the red pulp macrophages. This process is known as erythrophagocytosis, which is important for the turnover of red blood cells and the recycling of iron, which is a major function of the red pulp macrophages and is made possible by this special structure of the red pulp. The iron from the red blood cells is either released by the red pulp macrophages or stored in the erythrocyte itself in the form of ferritin. Also, the erythrocyte can store larger amounts of iron in the form of hemosiderin, an insoluble complex of partially degraded ferritin, and large deposits of this can be seen in the red pulp macrophages. The red pulp macrophages also obtain iron by scavenging a complex of hemoglobin, released from erythrocytes destroyed intravascularly throughout the body, and haptoglobin, via endocytosis through CD163. The iron stored in the splenic macrophages is released in accordance with the requirements of the bone marrow. Functions of spleen-specific and non-specific immune function, filter function, pooling of blood, reservoir function, hematopoiesis, humoral immune response, phagocytosis, involve in tissue healing. A lymph node, or lymph gland, is a kidney-shaped organ of the lymphatic system and the adaptive immune system. A large number of lymph nodes are linked throughout the body by the lymphatic vessels. In the lymphatic system, a lymph node is a secondary lymphoid organ. A lymph node is enclosed in a fibrous capsule and is made up of an outer cortex and an inner medulla.
Lymph nodes are kidney or oval-shaped and range in size from 2 mm to 25 mm on their long axis, with an average of 15 mm. Each lymph node is surrounded by a fibrous capsule, which extends inside a lymph node to form trabeculae. The substance of a lymph node is divided into the outer cortex and the inner medulla. These are rich with cells. The hilum is an indent on the concave surface of the lymph node where lymphatic vessels leave and blood vessels enter and leave. Lymph nodes are present throughout the body, are more concentrated near and within the trunk, and are divided into groups. There are about 450 lymph nodes in the adult. There are no lymph nodes in the central nervous system, which is separated from the body by the blood-brain barrier. Lymph from the meningeal lymphatic vessels in the CNS drains to the deep cervical lymph nodes. Lymph nodes contain lymphocytes, a type of white blood cell, and are primarily made up of B cells and T cells. B cells are mainly found in the outer cortex where they are clustered together as follicular B cells in lymphoid follicles, and T cells and dendritic cells are mainly found in the paracortex. There are fewer cells in the medulla than the cortex. The medulla contains plasma cells, as well as macrophages which are present within the medullary sinuses. As part of the reticular network, there are follicular dendritic cells in the B cell follicle and fibroblastic reticular cells in the T cell cortex. The reticular network provides structural support and a surface for adhesion of the dendritic cells, macrophages and lymphocytes. It also allows exchange of material with blood through the high endothelial venules and provides the growth and regulatory factors necessary for activation and maturation of immune cells. Lymph enters the convex side of a lymph node through multiple afferent lymphatic vessels, which form a network of lymphatic vessels, Latin plexus, and from here flows into a space, Latin sinus, underneath the capsule called the subcapsular sinus. From here, lymph flows into sinuses within the cortex. After passing through the cortex, lymph then collects in medullary sinuses. All of these sinuses drain into the efferent lymphatic vessels to exit the node at the hilum on the concave side. These are channels within the node lined by endothelial cells along with fibroblastic reticular cells, allowing for the smooth flow of lymph. The endothelium of the subcapsular sinus is continuous with that of the afferent lymph vessel and also with that of the similar sinuses flanking the trabeculae and within the cortex. These vessels are smaller and don't allow the passage of macrophages so that they remain contained to function within a lymph node. In the course of the lymphocytes may be activated as part of the adaptive immune response. A lymph node contains lymphoid tissue, i.e., a meshwork or fibers called reticulum with white blood cells enmeshed in it. The regions where there are few cells within the meshwork are known as lymph sinus. It is lined by reticular cells, fibroblasts and fixed macrophages. Thin reticular fibers, reticulin, of reticular connective tissue form a supporting meshwork inside the node. The lymph node capsule is composed of dense irregular connective tissue with some plain collagenous fibers, and a number of membranous processes or trabeculae extend from its internal surface. The trabeculae pass inward, radiating toward the center of the node, for about 1/3 or 1/4 of the space between the circumference and the center of the node. In some animals they are sufficiently well marked to divide the peripheral or cortical portion of the node into a number of compartments, nodules, but in humans this arrangement is not obvious. The larger trabeculae springing from the capsule break up into finer bands, and these interlace to form a meshwork in the central or medullary portion of the node. These trabecular spaces formed by the interlacing trabeculae contain the proper lymph node substance or lymphoid tissue. The node pulp does not, however, completely fill the spaces, but leaves between its outer margin and the enclosing trabeculae a channel or space of uniform width throughout. This is termed the subcapsular sinus, lymph path or lymph sinus. Running across it are a number of finer trabeculae of reticular fibers, mostly covered by ramifying cells. Peyer's patches, or aggregated lymphoid nodules, are organized lymphoid follicles, named after the 17th-century Swiss anatomist Johann Conrad Peyer. They are an important part of gut-associated lymphoid tissue usually found in humans in the lowest portion of the small intestine, mainly in the distal jejunum and the ileum, but also could be detected in the duodenum. Peyer's patches are observable as elongated thickenings of the intestinal mucosa measuring a few centimeters in length. About 100 are found in humans. Microscopically, Peyer's patches appear as oval or round lymphoid follicles, similar to lymph nodes. located in the mucosa layer of the ileum and extend into the submucosa layer. The number of Peyer's patches peaks at age 15 to 25 and then declines during adulthood. In the distal ileum, they are numerous and they form a lymphoid ring. At least 46% of Peyer's patches are concentrated in the distal 25 cm of ileum in humans. It is important to note that there are large variations in size, shape, and distribution of Peyer's patches from one individual to another one. In adults, B lymphocytes are seen to dominate the follicle's germinal centers. T lymphocytes are found in the zones between follicles. Among the mononuclear cells, CD4+, CD25+, 10% cells and CD8+, CD25+, 5% cells are more abundant in Peyer's patches than in the peripheral blood. Peyer's patches are characterized by the follicle-associated epithelium, Phi, which covers all lymphoid follicles. Phi differs from typical small intestinal villus epithelium. It has fewer goblet cells, therefore mucus layer is thinner, and it is also characterized by the presence of specialized M cells or microfold cells, which provide uptake and transport of antigens from lumen. Moreover, basal lamina of follicle-associated epithelium is more porous compared to intestinal villus. Finally, follicle-associated epithelium is less permeable for ions and macromolecules, basically due to higher expression of tight junction proteins. Because the lumen of the gastrointestinal tract is exposed to the external environment, much of it is populated with potentially pathogenic microorganisms. Peyer's patches thus establish their importance in the immune surveillance of the intestinal lumen and in facilitating production of the immune response within the mucosa. Pathogenic microorganisms and other antigens entering the intestinal tract encounter macrophages, dendritic cells, B lymphocytes, and T lymphocytes found in Peyer's patches, and other sites of gut-associated lymphoid tissue, GALT. Peyer's patches thus act for the gastrointestinal system much as the tonsils act for the respiratory system, trapping foreign particles, surveilling them, and destroying them. Peyer's patches are covered by a special follicle-associated epithelium that contains specialized cells called microfold cells, M cells, which sample antigen directly from the lumen and deliver it to antigen-presenting cells, located in a unique pocket-like structure on their basolateral side. Dendritic cells and macrophages can also directly sample the lumen by extending dendrites through transcellular M cell-specific pores. At the same time, the pericellular pathway of follicle-associated epithelium is closed tightly to prevent penetration of antigens and continuous contact with immune cells. T cells, B cells and memory cells are stimulated upon encountering antigen in Peyer's patches. These cells then pass to the mesenteric lymph nodes where the immune response is amplified. Activated lymphocytes pass into the bloodstream via the thoracic duct and travel to the gut where they carry out their final effector functions. The maturation of B lymphocytes takes place in the Peyer's patch. Gut-associated lymphoid tissue, GALT, is a component of the mucosa-associated lymphoid tissue, MALT, which works in the immune system to protect the body from invasion in the gut. Owing to its physiological function in food absorption, the mucosal surface is thin and acts as a permeable barrier to the interior of the body. Equally, its fragility and permeability creates vulnerability to infection and, in fact, the vast majority of the infectious agents invading the human body use this route. The functional importance of GALT in body's defense relies on its large population of plasma cells, which are antibody producers, whose number exceeds the number of plasma cells in spleen, lymph nodes and bone marrow combined. GALT makes up about 70% of the immune system by weight. Compromised GALT may significantly affect the strength of the immune system as a whole. The gut-associated lymphoid tissue lies throughout the intestine, covering an area of approximately 260 to 300 square meters. In order to increase the surface area for absorption, the intestinal mucosa is made up of finger-like projections, villi, covered by a monolayer of epithelial cells, which separates the GALT from the lumen intestine and its contents. These epithelial cells are covered by a layer of glycocalyx on their luminal surface so as to protect cells from the acid pH. New epithelial cells derived from stem cells are constantly produced on the bottom of the intestinal glands, regenerating the epithelium. Epithelial cell turnover time is less than 1 week. Although in these crypts conventional enterocytes are the dominant type of cells, Paneth cells can also be found. These are located at the bottom of the crypts and release a number of antibacterial substances, among them lysozyme, and are thought to be involved in the control of infections. Underneath them, there is an underlying layer of loose connective tissue called lamina propria. There is also lymphatic circulation through the tissue connected to the mesenteric lymph nodes. Both Galt and mesenteric lymph nodes are sites where the immune response is started due to the presence of immune cells through the epithelial cells and the lamina propria. The Galt also includes the Peyer's patches of the small intestine. Isolated lymphoid follicles present throughout the intestine and the appendix in humans. Galt can be also divided into 2 categories considering the structure, from which the function arise. There can be found 1. Organized Galt made up from follicules, such as Peyer's patches, mesenteric lymph nodes and even more organized appendix. Its main function is to induce immune reaction. 2. Diffuse Galt with single T and B cells, macrophages, eosinophiles, basophiles and mast cells, preferentially found in lamina propria. This part of Galt is made up from mature effector cells ready to perform their actions. The mucosa-associated lymphoid tissue, MALT, also called mucosa-associated lymphatic tissue, is a diffuse system of small concentrations of lymphoid tissue found in various submucosal membrane sites of the body, such as the gastrointestinal tract, nasopharynx, thyroid, breast, lung, salivary glands, eye, and skin. MALT is populated by lymphocytes such as T cells and B cells, as well as plasma cells and macrophages, each of which is well situated to encounter antigens passing through the mucosal epithelium. In the case of intestinal MALT, M cells are also present, which sample antigen from the lumen and deliver it to the lymphoid tissue. MALT constitute about 50% of the lymphoid tissue in human body. Immune responses that occur at mucous membranes are studied by mucosal immunology. MALT plays a role in regulating mucosal immunity. The components of MALT are sometimes subdivided into the following. GALT, gut-associated lymphoid tissue. Peyer's patches are a component of GALT found in the lining of the small intestines. BALT, bronchus-associated lymphoid tissue. NALT, nasal-associated lymphoid tissue. CALT, conjunctival-associated lymphoid tissue. LALT, larynx-associated lymphoid tissue. SALT, skin-associated lymphoid tissue. VALT, vulvo-vaginal-associated lymphoid tissue. TALT, testis-associated lymphoid tissue. It can be also distinguished by level of organization of the tissue. OMALT, organized mucosa-associated lymphatic tissue. Specifically, the tonsils of Waldeyer's tonsillar ring are OMALT. DMALT, diffuse mucosa-associated lymphatic tissue. MALT that is not organized as a separately macroscopically anatomically identifiable mass, tissue or organ, such as the aforementioned OMALT, is diffuse MALT.